Good evening, guys. Tonight's discussion C deals with three major points that I want to get across. So here we go. A major takeaway from a recent study, Super Agers research, and is that these unusually high-performing older adults show distinct brain, cellular and genetic features that may point toward new Alzheimer's prevention and treatment strategies. Across multiple studies, scientists have identified biological signatures ranging from enhanced neuron generation to resilient brain networks and protective gene variants that could help explain why some people maintain youthful memory into their 80s and beyond. National Institutes of Health, NIH, Medical Express, WUTSAI, Neuroscience Institute, what the studies found: enhanced neurons creation, super agers hippocampi contain more newly formed neurons and unique molecular signatures linked to neuron development. These signatures differ even from cognitively healthy older adults and contrast sharply with the impaired neuron generation patterns seen in Alzheimer's disease. This suggests that supporting adults' neurogenesis could be a therapeutic pathway. Protective genetic profile. Large-scale genetic studies show superagers are far less likely to carry the high-risk APOE4 allele and more likely to carry the protective APOE2 variant. These genetic patterns may help identify biological mechanisms that counter resilience to Alzheimer's pathology. Resilient brain networks. Functional MRI research has identified an Alzheimer's resilient connexome arc, a connectivity pattern in prefrontal temporal and insular networks that distinguishes super agers from Alzheimer's patients with acuity or high accuracy, individuals with an arc-like profile show slower cognitive decline and reduced amyloid accumulation, making this a promising early biomarker for risk satisfaction and targeted prevention. Long-term brain resilience. Over 25 years of research, superagers consistently show resistance to Alzheimer's related plaques and tangles, along with lifestyle traits such as strong social engagement. Their brains reveal structural and cellular features that challenge the assumption that cognitive decline is inevitable. Why this matters for Alzheimer's treatments. Together, these findings highlight several promising therapeutic directions. Boosting adult neurogenesis to mimic the cellular environment seen in superagers. Developing treatments targeting ARC like network prevention, potentially slowing or preventing cognitive decline using genetic markers. APOE2, CLYBL, FRMD6 identify individuals most likely to benefit from early interventions. Designing precision prevention strategies that combine biomarkers, genetics, and brain network profiling. These converting lines of evidence position superagers as a powerful model for understanding brain resilience and for guiding the next generation of Alzheimer's therapies. The study functional connect zone of superagers reveals early markers of resilience and vulnerability to Alzheimer's disease. Identifies a distinctive brain network signature called the Alzheimer's Resilient Connect Zone or ARC that separates superagers from individuals with Alzheimer's disease and predicts who among cognitively normal adults is most likely to remain resilient or become vulnerable to future decline. Alzheimer's resilient connectome arc using resting state MRI and machine learning classifiers, researchers identified a functional connectivity pattern across prefrontal, temporal, and insular networks that distinguishes super agers from age-matched Alzheimer's patients with higher acuity or accuracy. AUC equals 0.85. Predictive power in healthy adults. When applied to large samples of cognitively unimpaired adults, ARC separated them into SA-like and AD-like subgroups. So, who had similar baseline cognition but dramatically different aging trajectories. SA-like individuals showed slower cognitive decline, reduced amyloid accumulation, lower risk of processing to MCI or Alzheimer's disease. Genetic modulators. Genome-wide association analysis identified CLYBL and FRMD6 as genes linked to these divergent resilience versus vulnerability patterns. Why this matters for Alzheimer's research. The ARC signature functions as a sensitive generalizability biomarker that can identify resilient versus vulnerable individuals before symptoms appear, enable early risk satisfaction, support precision prevention strategies, targeting network level brain aging. Guide development of interventions that preserve or mimic super ager-like connectivity patterns, how these fit into the broader super ager literature. Other studies show mixed results about whether functional connectivity alone explains super aging, suggesting that AR's arc may capture more specific disease relevant network signatures rather than general connectivity strength. Complementary research also finds unique cellular signatures and enhanced adult neurogenesis in superagers, reinforcing the idea that resilience emerges from multiple biological layers. Okay, guys, now that is it for tonight. I will be back on tomorrow for shows. Until then, Gator Wellness. Take care, folks. Have a good evening. See you tomorrow.